Okay. Hi everyone, my name is Jessalyn. I am one of your Bio5a SIs. And today we're going to be going over translation. If you don't know what transcription is, I would encourage you to watch Jonathan's video. It's called Week 9 Video 1 on transcription, and then we will be talking about translation in this video today. So first of all, what happens before translation can begin? We have transcription that creates the mRNA, right? But within this mRNA, there's your exons and your non-coding introns. So the first thing you have to do is remove the non-coding introns. Then you have to add the 5' cap and the 3' poly A tail. And this occurs via the spliceosome. And so once you do that, you have your mature mRNA, which leaves through the nuclear pore and enters the cytoplasm to begin translation. Just remember, transcription is in the nucleus, translation is in the cytoplasm. So just a quick review on how to read codons. You're going to read from the 5' to 3' direction. So we say the start codon is AUG, you're reading from 5' to 3'. Same with the stop codons, UAA, UAG, and UGA. Okay, so for translation, it is carried out by an assembly line. You have your ribosome, which is your factory, and your ribosome is a large multi-subunit enzyme containing both RNA and proteins. And then you have your tRNA, so right here, your tRNA, these are your workers, and they are bringing the amino acids over. Just to reiterate, your 5' to 3' direction of the mRNA, right? So if we read AUG, that's how we read it in this direction. Okay, then your transfer RNA, so tRNAs, are linked triplet codons to amino acids. So this is the shape of your tRNA. It's literally the shape of a T, so that's easy to remember. Within all of this, right, these are all hydrogen bonds, keeping the shape of the tRNA. So all these little lines here. Then you have your amino acid up here that is attached to the 3' end. This is by a covalent bond right here. So hydrogen bonds here, covalent bond there. And then you have your nitrogenous bases that are paired together, right? So on your mRNA strand, you have your codon, while your tRNA carries your anticodon. Okay, so tRNAs are activated by enzymes. And so the enzyme we're using is amino acyl tRNA synthetase. There are 20 different amino acids, right? So there's 20 different enzymes, so they are each specific. And so that specific amino acid hops into this area, as well as ATP, right? So once that happens, a pyrophosphate leaves, and there's a phosphate group attached to this amino acid. Then your tRNA comes in, your phosphate leaves, so your AMP here, and then the amino acid binds to the tRNA. So this is called your charged or activated tRNA, and that's how you form how that tRNA comes in and brings that amino acid, and you form your polypeptide chain. And we'll go in more detail as we go on with the slides. Okay, so when we look at the ribosome, there are 3 sites, right? There's the A site, which is, sometimes they call it your receptor site, but it's your amino acyl tRNA binding site over here. Then you have your P site, which is your peptidyl tRNA site. This is where your polypeptide is gonna grow. So when you have like a tRNA here, you're gonna have a chain of amino acids. So that's just gonna form a long chain, and then once it reaches a stop codon, then it will end. And then your E site is the exit site, and this is where just the tRNA is here, there is no amino acid, and it'll just leave the ribosome. And so over here is your unit. You have your small subunit. This one attaches to the mRNA first, and then your large subunit comes in with the tRNAs, bringing in the amino acids. Okay, the 3 steps of translation. First one is initiation. So let's look here. You have your small ribosomal subunit, right? It binds to the 5' end over here. Then it's going to slowly scan the mRNA. So as it reaches here, it's going to slowly go down towards this way, towards the 3' direction to find the start codon, or to find AUG. Once it reaches AUG right here, the tRNA is already the UAC and the AUG match, right? So then it tells the large ribosomal subunit to come in. Once that comes in, and it requires GTP, so you require energy for this process, this whole thing is your initiation complex. So now you can start translation. Once you have reached the start codon, now all the tRNAs can come in and find the respective codons and like match that as well. So we have elongation. So 3 steps of elongation, and it's a cycle. Number 1 is codon recognition. So the tRNA bringing the matching codon. So over here you see GTP is required. So the tRNA is bringing in the next matching codon with the amino acid. Then you have your peptide bond formation, right? So your new peptide bond is when the amino acid over here, it jumps over and bonds to the other amino acid. And remember when we learned about the 4 macromolecules? You learned about amino acids and how they form peptide bonds with each other through dehydration synthesis. So this is the same thing, you're forming that polypeptide, that peptide bond. Then you have translocation, which is the tRNAs moving from the A and P site to the P and E site, and the large subunit moving over. So this whole shift also requires GTP, or requires energy. Main thing you have to remember for translocation, you have to free up the A site because this is the receptor site, right? So a tRNA has to come through the A site and it'll move over to P and then E where it'll exit. So again, GTP required for matching codons, and then translocation as well, GTP is required. Okay, last step of translation, you have termination. And this happens when you see a stop codon, right? So this codon on your mRNA is your stop codon. It can be UAG, UAA, or UGA. And it triggers this release factor, which is a protein, and it'll come and come through the A site and bind to the stop codon. Then it tells the tRNA at the P site to leave, and it will free this polypeptide. So this is what translation is about, right? We want this polypeptide because it's going to form a lot of functions. Once that leaves, you have GTP that triggers everything to fall apart. So your large and small subunits are going to fall apart, release factor goes away, and your mRNA goes away. So everything breaks apart, and that initiation complex you had at first is now all broken, and you don't have that anymore. Okay. The Wobble Effect. So this is actually something that's really interesting, because if you look at the amino acid chart, the one where all the codons match with a certain amino acid, you know that a lot of the third base pair of each codon doesn't really matter, because it can code for the same amino acid. For example, LEU, right? Same amino acid. So GC, that's a normal pairing, right? Because those two go together. However, you can have a wobble pairing, which is incorrect, right? G and U don't belong together. But in mRNA, it has more tolerance. You have tolerance of, the wrong pairing, but you still get the same amino acid because it codes for the same thing. So this is called your wobble effect, where your third base pair has more tolerance. Okay, so we're gonna switch on to prokaryotic translation. Real quick, polyribosomes, these are multiple ribosomes translating the same RNA simultaneously. So this doesn't happen in eukaryotes, but it happens in prokaryotes because transcription and translation happen at the same time, and you can have a lot of translation sites happening, right? So you look like your pink is your mRNA, and you have all these different units together forming polypeptides. So your purple is your polypeptide leaving. Yeah, so we're going to go over ribosomes really briefly. There is your free ribosomes, which are in the cytoplasm, and you have your bounded ones, which are on the ER proteins. And so the proteins of the free ones are targeted to chloroplasts, mitochondria, and the nucleus. And then your bound ribosomes, it targets a new protein to the endomembrane system. So over here, right, these are all your endomembrane system. Okay, so that is it. Thank you guys for watching this video. If you have any questions, I will list my email in the comment section, and thank you very much.